So let us continue with the session on gene regulation. As we discussed earlier, there are different forms of gene regulation which may affect in different stages of gene expression, starting from the transcription and finally which ends at the protein degradation. So there are various stages in gene expression, and at each, in between each and every stage we can expect one or another form of a regulatory mechanism or checkpoint that will be working in controlling the overall gene expression. So the first one that we mentioned is about histone modification and other DNA methylation mechanisms, which will be working at the level of chromatin rearrangement, or it is the first at the initial stage of the gene expression. So in this picture we can see that there is a chromatin change. That means the condensed chromosome is unwound and the free chromatins were now made available for transcription machinery. Then transcription occurs, RNA processing, splicing mechanisms, all these things will happen. Then after the RNA processing, the processed mRNA may be subjected to degradation without going for translation, or it may be translated and subsequently the translated protein may be modified or degraded, or it may be used for the cellular activities. So in between each and every stage, we can have some form of control or regulation of the overall gene expression process. In the first one, as we mentioned, we have the regulation of a chromatin structure. In the chromatin structure, as we know, the chromatins were actually wound over the histone proteins, and here a unit of histone protein is shown, which is wound around by the chromatin, and here it is the histone proteins and the chromatin or DNA, which is wound around that structure. Then DNA double helix is marked here. So these are the amino acids of the histone proteins that were available or to the exterior surface for chemical modification. So these amino acid tails or the histone tails were modified by basically by acetylation. So acetyl groups were added to these amino acids that is available through the exterior portion of the histone proteins, and these were acetylated. So that is one of the histone modification which will result in a structural change and the release of the DNA helix from the histone complex, so that the free chromatin will be made available for a transcription. So that is the histone tails are there, which will be linked with which will be modified, chemically modified with the acetyl groups, and that is the acetylation modification of the histone tails. Now, here is the histone compact unacetylated histones. Histone proteins were tightly packed with the DNA. Now, after the acetylation process, that will become a loose network, and acetylated histones will unwind, or that condensed chromosome structure will unwind, and the DNA strands will be made available for transcription machinery to act on. And another one is the DNA methylation, and that DNA methylation can inactivate the genes and be inherited by offsprings. Genomic imprinting works in this way. So that means this DNA methylation also inactivates some genes, and methylated DNA can be transferred to the offsprings also, so that these DNA imprinting or genomic imprinting can be transferred to the offsprings in this manner. So this is about the different chemical modifications occurring at the initial stage of the gene expression, and that is actually the modification of acetylation and methylation to the histone proteins, which enables that histone proteins to change its structure, and the compact structure of the chromosome will be unwound, and that chromatin will be made available for a further transcription process. Okay. Now, here is a comparison of prokaryotic and eukaryotic gene expression. You see, the small genome is present in case of prokaryotes, whereas in case of a eukaryote, we have a very large genome. And specialization or cell differentiation is not present in prokaryotes, whereas cell specialization or cell differentiation into different specific cell types like neuronal cells or some other bone marrow cells or like muscle cells, all these things will be different types of islets of Langerhans means in the pancreatic cells, lens cells. All these are different types of cells with a specific functions, and that specialization or the cellular specialization or cell differentiation is occurring only in case of eukaryotes and not in prokaryotes. And most of the DNA codes for proteins or RNAs in case of prokaryotes. That means there is only very little junk DNA or non-coding regions which is present in prokaryotes, whereas most of the DNA in eukaryotes doesn't code for anything, or it doesn't code for proteins or RNAs or some functional components. So that means junk DNA is almost 98% in case of such a eukaryotic systems, especially in humans. And genome, actually it is in prokaryotic genome, it is a DNA plus a few proteins in a simple arrangement, whereas in the genome of the eukaryotes is DNA with a large number of proteins in complex arrangements. So that means the histone proteins and other supporting proteins were interacting with the genome of the eukaryote, which is not present in prokaryotic systems. Then RNA processing is not an option for controlling gene expression in case of prokaryotes, whereas RNA processing is a major phenomenon that is occurring in the eukaryotic gene expression, and there are control elements or gene control or gene regulation possibilities are there at that stage in case of eukaryotes. So RNA processing allows for several opportunities to regulate the genes in eukaryotic systems. Then mRNA has a short lifespan of a few minutes, 1 or 2 minutes in case of prokaryotes, whereas the eukaryotic mRNAs were long-lived. Long-lived means maybe days to months of lifespan is there for eukaryotic mRNAs. So their regulation mechanism is entirely different from that of the prokaryotic systems. So both eukaryotes and prokaryotes, their gene expression in response to the environment is there. There is gene expression in response to environment is there, and the transcription initiation is the most important control point in both eukaryotes and prokaryotic systems. So they are actually, their gene expression is well regulated by the environmental, internal or external environmental factors which stimulates the gene expression, and there are various control points at the initiation of transcription in both the eukaryotes and the prokaryotes. Now this is about cell-specific gene transcription. Sorry. Here we have two different cell types, that is liver cell and lens cell is given. So there are same control elements with the enhancer. In eukaryotes there are various elements or enhancer and activator elements which is present upstream, and the enhancer elements or the control elements were in both the genes. This is the albumin gene and this is the crystalline gene. Albumin gene is present in liver cells and crystalline gene is present in lens cells of the eye. And here we have the promoter for both, and this is the gene. Both liver cells and the lens cells have the genes for making the protein, albumin and crystallin. That means in both cells we have the same gene because it is the same organism, it contains the same genome. So both the cells is having both the genes. But only liver cell makes albumin, that is a blood protein, and only lens cell makes crystallin, the main component of the lens of the eye. So these specific transcription factors or the activators and repressors, they are the transcription factors made in a particular type of the cell determine which genes are expressed. So the transcription factors that is acting on liver cell will not be produced in lens cells, and those transcription factors acting on lens cells will not be produced in liver cells. So based on the response of the transcription factors, that is the activators and repressors, the genes will be expressed differentially in two different cell types. In this example, the genes of the albumin and crystallin are shown at the top, and each with the enhancer made up of three different control elements. So this is the three different control elements for the genes. Although the enhancers for the two genes share one control element, each enhancer has a unique combination of elements. So each enhancer has a unique combination of elements, and they will be interacting with a different set of activator or repressor or regulatory proteins or transcription factors. So these different control elements will interact with the different proteins, that is a transcription factors or activators or repressors, and the result is that the genes will be expressed differentially. Okay, although the enhancers for two genes share one control element, each enhancer has a unique combination of elements, and all the activators required for high level expression of the albumin gene are present only in liver cells, whereas the activators needed for the expression of crystallin gene are present only in lens cells. For simplicity, we consider only the role of activators here, although there are presence of repressors, that they are the other transcription factors which is present. So activators and repressors, all these things will interact differentially in these two different cell types, resulting in two types of different cell expression or gene expression in two different cell types. So this is one of the classical example of a cell-specific gene expression, and you can see liver cells, we have albumin gene specifically expressed, and in lens cells we have crystallin gene specifically expressed, and in maybe in pancreatic cells we have insulin gene specifically expressed. These are all cell-specific expression based on their specific control elements to which the specific cell-specific transcription factors, that is the activators or repressors, will interact and resulting in the control in the specific type of a cell gene expression. So this is a cell-specific transcription or cell-specific expression of the genes. Okay, now this is about the alternative promoter utilization. Alternative promoter utilization, we discussed this thing during the alternative splicing, where we have different promoter elements, and if the promoter for the liver, so the same genes is present in the same cell, but in liver cell and B cell we have the same genes which is present, or the same control elements are present, but the transcription factors will bind to specific combination of the promoter regions, resulting in a specific expression. So you can see that in liver cells we have some set of elements which is acting in combination, whereas in B cells another set of elements which is acting in combination as the regulatory element. So in both the systems we will have cell specific activators that will bind to that specific regions, resulting in a different gene expression. So now we already discussed the chromatin change or the histone protein modifications and some little bit about the transcription level regulation. Now at the transcription level regulation and RNA processing, this is the overall process that is occurring. And we know that this is the typical eukaryotic gene in which we have some enhancers or distal control elements or which is present upstream or before the start of the gene, we will have upstream elements, upstream activator sequences or enhancer sequences or distal control elements. All these things are the different regulatory elements which is present in the eukaryotic gene. And these are proximal control elements which will be present near the promoter region, and this is the promoter region. Now these are the different exons, the introns in between the exons, and finally there is a poly A signal sequence, and which will be recognized by the splicing system, and subsequently it is removed at this point. The excess region downstream will be removed from this point, and poly A tail will be added by recognizing the poly A signal, and there is a termination sequence which will be used for termination of transcription, which is present downstream of the gene. So once this gene eukaryotic gene is expressed, the transcription occurs and the primary transcript or the pre-mRNA is produced, which will have a 5' end and different exons and introns. Then finally there is a poly A signal and there is a 3' end which needs to be spliced off. And finally the RNA processing occurs, which includes capping and poly A tail addition, intron removal, and exon splicing or joining together of the exons, and resulting in the final processed mRNA, which will have a 5' cap, 5' methyl cap, then upstream UTR untranslated regions, maybe that is some regulatory elements. Then we have start and stop codons, then 3' UTRs or untranslated regions, then poly A tail. So this is the actual coding segment in between the start and stop codons, and this region will be translated to produce a functional protein. So this is a typical structure of a eukaryotic mRNA. Now we will discuss this in another slide also, the lac operon concept. So the lac operon is the best example of the gene regulation, or whenever we are learning gene expression in prokaryotic system, we have the typical example of lac operon. And we will discuss the operon concept in another session, and the basic concepts will be discussed at that point. Here, as you know, the lac operon is having a cap binding site or positive regulation element is there. Now there is a RNA polymerase binding site, or that is a promoter and operator regions are there. There is a promoter region, this is the operator region, and these are the lac Z gene. So starting from this point, the RNA synthesis starts. So now we will have different conditions like glucose present or glucose absent or lactose present or lactose absent, and in response to that the operon will be turned on or off. Okay, finally only at a very specific condition like glucose absent and a lactose present, the RNA will be produced, or the operon is expressed only at a specific condition. So according to this concept, lac operon concept, both positive and negative regulation mechanisms will be present in case of lac operon, and it recognizes these external stimuli of the presence of lactose and absence of glucose using these positive and negative regulation mechanisms. Then finally the RNA transcript is produced in the case of lac operon. This is one of the examples of the regulation of the gene expression at the transcription level, and we will discuss the operon concept later. So we are not going into the in depth into the lac operon concept, that can be addressed later. But here is an example. Lac operon is an example of a regulation of gene expression using positive and negative mechanisms at the transcription level. Okay. So here is another one. This is a trip operon, and which also we will discuss later. So we have various control elements, and during the transcription process itself, these trip operon will regulate the production of the concerned mRNAs based on the availability or requirement of the tryptophan in the medium. So if the tryptophan is present, biosynthesis is not required, so the operon will shut down. And if tryptophan is absent, needs to be biosynthesized, then it will recognize that requirement from the system, and it will start coding for the concerned mRNA. So this is another example of a transcription level regulation of gene expression. And in trypoperon, we have this attenuation system, and that we'll discuss in another slide. Attenuation system. Okay. Then prokaryotes have operons to control expression of the genes and related functions. What about eukaryotes? Functionally related genes are co-expressed because they have the same control elements that are activated by the same chemical agents. So usually in prokaryotic systems, all the required genes were placed under the control of a single promoter or single regulatory element, and they are termed as operons. Okay, all the required mRNAs will be simultaneously coded by this starting from a single regulatory element. Whereas in prokaryotes, eukaryotes, we don't have such an operon system, because even the single gene is interrupted by the intron regions. That needs to be spliced, or splicing mechanism is effective in case of eukaryotic genes. But in eukaryotes also, there is a phenomenon of a co-expression of similar genes or related genes, or the genes that is required for achieving a common goal. Related genes which needs to be coded together will be placed in the immediate vicinity and maybe under the control of a same regulatory element or control element, so that they will be transcribed simultaneously. So it is not the concept of the operon, which is simultaneously coded as a single mRNA transcript, but the regulatory element may be same, but they are actually coded and spliced separately in case of eukaryotes. So it is not an operon, but such genes, eukaryotic genes which needs to be co-expressed, were placed under the control of a same regulatory element, and they will be independently transcribed. Whereas in case of operon, we have a single regulatory element or a promoter region or an operator region, and the mRNA will be transcribed in a single stretch, which needs to be translated from that single primary transcript. The concerned genes or the cluster of the genes in operon system needs to be translated from the single primary transcript. Whereas it is not the case of eukaryotic genes. There is no operons, and the related genes were placed under the control of a same control element, and the genes will be expressed separately starting from the control element. So here we have another example of another type of gene regulation. So this is the SOS response in Escherichia coli, and here the Lexe protein, this is a repressor in this system. Here is the Lexe repressor protein, the red ones, and it actually binds near every gene. Because the Recce gene is not entirely repressed by the Lexe repressor, so this is the Recce gene, and this is not completely repressed, whereas the other genes in the system were completely repressed by the Lexe repressor protein. Whereas this gene, Recce gene, is not completely repressed. Even though it is repressed, it is not completely repressed, and some amount of Recce protein can be produced even after binding of this Lexe at this point. Okay. Now, Because the Recce gene is not entirely repressed by the Lexay repressor, the normal cell contains about 1,000 Recce monomers. So they are present here. When the DNA is extensively damaged means the DNA will be single stranded at that point, because extensive damage prevents the duplex formation. So single stranded gaps will be there, and DNA that gaps will be actually, it is a broken part or a thymine dimer or some other structural irregularities will be there at such a damaged positions, which will leave the strands as a single strand. Then when it is extensively damaged, the DNA replication is halted and the number of single strand gaps in the DNA increases. Then Recce protein binds to the damaged single stranded DNA. This protein binds to the single stranded region of the DNA, activating the protein coprotease activity. So this Recce protein is having a coprotease activity. So when it binds to the single stranded region of the DNA, this coprotease activity of the Recce protein is activated, and that coprotease activity will cleave the Lexay repressor proteins available here. So when the Lexay proteins is cleaved by or removed by the Recce coprotease activity, then all the other cluster of the genes which is present here, that is a that genes will be that SOS response or which will be a response protein for in response to the extensive damage of the DNA. That set of the genes will be expressed once the Lexay is cleaved and removed from the repression system. Now, while bound to the DNA, the Recce protein facilitates cleavage and inactivation of the repressor, Lexay repressor. And when the repressor is inactivated, the SOS genes, including the Recce, all these Recce genes, all this will be coded, and sufficient quantity of the Recce and the other repair enzymes will be coded by the system. And if there is no or once all the gaps were filled and the repair is made, then this will be single stranded. There will not be binding of the Recce protein at this point, and the cleavage activity of the or the protease activity will not be available in the system. So that means all the Lexay proteins, which is or the repressor proteins that is produced by the Lexay, will bind to the concerned genes, and now it is it blocks the entire gene expression system. So this is the SOS repair in E. coli that uses the Lexay repressor protein and Recce protein. So this is one of the examples of a gene regulation in E. coli, that is the SOS response of the E. coli. So these Recce levels increase 50 to 100 folds. So normally in normal systems, Recce is only expressed at a very low level even after binding of the Recce protein. Then when the Recce protein or this inhibitor or Lexay protein is removed from the Recce system, about 50 to 100 fold increase in the Lexay in the Recce protein will be there. Then that will bind, that can bind and further in all other regions of the damaged DNA and facilitate or the repair SOS response will be enhanced by the additional Recce proteins that is produced. So once the DNA is completely repaired, then there is no need for binding of the Recce protein, and Lexay proteins will bind back to the concerned genes or the SOS response genes. Then it will come back to the normal position. So this is about the SOS response in E. coli. Now this is about gene regulation by recombination. This is another mechanism, and here we have another example of Hin recombinase. Hin recombinase is a Here we see that there is a inverted repeat. There are two yellow inverted repeats which is known as Higgs regions in this gene. And there is a Hind recombinase enzyme which is coded from this gene. This Hind recombinase produces the mRNA and Hind recombinase is coded. This is the Hind recombinase is the enzyme that facilitates the recombination of this region in between these. So it facilitates recombination. So this inverted repeats region, this Higgs region will be, or the segment in between this Higgs region will be inverted by the effect of a Hind recombinase. So just like an inversion process between these two Higgs regions can be made, it can be done by the Hind recombinase enzyme which is coded by this Hind gene. Now, here is a promoter, FLJB promoter is there. So starting from this point, normally an mRNA can be coded, FLJB and FLJA. An mRNA can be coded if the promoter is in the correct orientation. So starting from this direction, if the promoter is in this particular direction, it starts the transcription and the mRNA will be coded. Both your FLJB and FLJ, that is a flagellin gene B and flagellin gene A, both will be coded simultaneously. And there is another promoter, a separate promoter of FLIC, that is a flagellin type C promoter is there, which is a separate gene. So what happens is that if this gene is coded, FLGB flagellin is coded, and the FLGA protein, it is not a flagellin protein, but it's a repressor protein for this promoter. FLC promoter repressor is coded by this FLJA. So if this gene is coded, for that the promoter of FLJB is to be in this direction. So if it is in the correct direction, both the genes will be coded. Along with the FLJB flagellin, the FLJA repressor which is required to block the coding of FLIC is also coded, and it will block the transcription of FLIC. That means only flagellin B, type B, is coded in this system. A normal case, the promoter orientation is in this direction, which codes the single mRNA. Both the FLJB and FLJA will be coded. FLJB is the flagellin, FLJA is the repressor for the C, and it binds to C, and C is no longer available in this normal system. But the Hind recombinase which is produced here, this will work on this gene, and an inversion between this sequence is done. So if this segment between the yellow Higgs genes is inverted, the promoter of FLJB will have a separate orientation, and it tries to start the coding of the mRNA in this direction and not in the reverse direction. So this will be a nonsense mRNA, or it is not the required flagellin gene. So in effect, this FLJB and FLJA is not coded in here. Whereas starting from this gene, the Hind mRNA can be again coded, and Hind recombinase will be again produced. Okay. So without the flagellin repressor A, the FLIC, that is a flagellin type C, will be coded. So the regulation mechanism ensures that there is both, in both the direction we have the Hind recombinase, or the recombinant enzyme is coded, and only one type of flagellin, that is flagellin A, flagellin B or flagellin C, any of them is coded, or only one of them is coded at one time. If flagellin B is coded, along with that, the repressor is also coded, blocking the coding of flagellin C. And if it is inversed, or the Hind recombinase inverse the direction of the promoter, then FLJB and the repressor both will not be coded, and the FLIC will be coded, and the flagellin type C will be expressed. So this is how the regulation by recombination is occurring in prokaryotes. Okay, so this is one of the examples of gene regulation by recombination.